The overall goal of the following experiment is to demonstrate shallow doping of silicon wafers and nanowires by using the monolayer contact doping method. This is achieved by reacting a donor substrate with a precursor solution for the formation of a self-assembled monolayer. The monolayer contains potential dopant atoms and serves as a well-defined and limited dopant source. As a second step, the donor substrate is brought into contact with the target substrate, and both are annealed in a rapid thermal anneal system. During the anneal process, the monolayer molecules decompose, and the dopant atoms are diffused and activated onto both the donor and target substrates. Next, the two substrates are separated, and a 4-point probe setup is used for measuring sheet resistance. Results show typical decrease in sheet resistance values measured for the target substrates annealed for various anneal times and temperatures.
The main advantage of this technique over existing methods like ion implantation is that it enables simple yet reliable formation of very shallow doping profiles.
This method is useful for surface doping of all wafers as well as nanostructures. It can also be used to other systems such as 3D device architectures and more.
Identify the silicon and the silicon with dielectric substrates that will be used as target and donor. Prepare to work with Piranha solutions. Acquire enough acidic Piranha solution of hydrogen peroxide and sulfuric acid to cover the substrates, here about 120 milliliters. Use extreme caution while working with the Piranha solution. Place the substrates in an appropriate holder. Insert